A virus that's mastered the art of breaking into human cells, except for a tiny group of people who are basically unhackable. Here's the thing. HIV normally uses a door on your immune cells called CCR5. That's its entry key. But some humans are born with a mutation called CCR5-32, which literally deletes a chunk of that door. No door, no entry, no infection. Virus stands outside like an Amazon package in the rain. Only 1% of people globally have 2 copies of this mutation. These are the superheroes, completely resistant. Another 10% have 1 copy. They still can get HIV, but the virus struggles like it's lagging on a 2G network. What this really means is our immunity isn't just biology, it's code. And sometimes there's a glitch in that code that flips you from vulnerable human to immune outlier. Scientists noticed this mutation was super common in Europeans whose ancestors survived the Black Death. Some believe the mutation spread because it protected people from ancient plagues, the original natural selection DLC. And now researchers are trying to copy-paste this mutation into others using CRISPR, basically turning normal immune cells into the Delta 32 edition, a real-world cheat code, born in 1% of people, now being reverse-engineered for the entire planet.
Is it possible to cure the incurable? I mean, really cure it? How do you fight a disease that literally rewrites itself into a person's DNA? Well, we're about to dive into one of the most incredible medical detective stories you've ever heard. You see, the big problem with HIV is that it's not just some bug passing through. It actually gets into your DNA. It hijacks your own immune cells and turns them into these little virus-making factories. So how on earth do you beat something like that? For the longest time, the answer was, you don't. It was impossible. Until one man, Timothy Ray Brown, completely changed the game. And his story, it sounds like something out of science fiction. But here's the thing. He wasn't in some cutting-edge experimental trial. He was just a guy fighting for his life against 2 different deadly diseases. And in a wild twist of fate, the treatment for one of them, well, it just so happened to cure the other. Okay, so check out this timeline. It's just wild. In '95, he gets diagnosed with HIV. Then, about 10 years later, bam, he gets hit with acute myeloid leukemia, a really aggressive blood cancer. Now, to treat the leukemia, doctors had to do something extreme. They had to basically destroy his entire immune system and then rebuild it with a stem cell transplant from a donor. They actually had to do it twice. And after all that, after this incredibly tough procedure, something happened that nobody expected. The HIV, it was gone. And I don't mean suppressed or dormant, I mean gone. He became the first person ever to be cured of HIV. So here's where it gets really interesting. It turns out the cure wasn't the cancer treatment itself. See, his doctor in Germany had this absolutely brilliant idea. It was a total long shot. He thought, what if I could find a stem cell donor who's not just a match, but who is also naturally immune to HIV? That was it. The secret wasn't the transplant. It was locked away in the donor's DNA. So what is this genetic secret? How does that even work? Well, to get it, you got to understand how HIV breaks into our cells. Think of it like a burglar. The virus needs a way in, like a door or a window. It needs to pick a lock. But for a very, very small number of people on Earth, that lock, it just doesn't exist. Okay, so this lock is actually a protein on the surface of your immune cells. It's called the CCR5 receptor, but let's just call it the doorknob. For the most common types of HIV, this doorknob is its golden ticket. The virus just latches on, turns the handle, and waltzes right inside to start causing chaos. And this visual just nails it. On one side, you've got a normal immune cell covered in these little doorknobs, the CCR5 receptors. It's an open invitation for HIV. But on the other side, you see that? The doorknobs are gone. A rare genetic glitch means the cell never even bothers to install them. So when HIV comes knocking, it's like running headfirst into a brick wall. There is no way in, period. And the science here is super clear. To get this biological superpower, you have to get a copy of this specific genetic mutation from both of your parents. That's what being homozygous means. If you've got that, you're basically bulletproof against most HIV strains. And Timothy Ray Brown's donor? he was one of those lucky few. So when they gave Timothy his stem cells, they were essentially giving him a brand new immune system, one built from a genetic blueprint that HIV just could not hack. So let's zoom in, way down to the genetic level. The instructions for building that CCR5 doorknob are on a gene located on our third chromosome. The mutation that changes everything is called delta 32. And really, it's just a tiny piece of code that's been deleted. Just 32 letters of DNA missing. It's a tiny mistake, but the result is huge. The protein that gets built from these broken instructions is basically a dud. It's misshapen, non-functional, and it never even gets installed on the outside of the cell. No doorknob. But this whole thing raises a really deep question, right? HIV is a pretty modern virus. We only really identified it back in the 80s. So why would a natural defense against it even exist? Where did this genetic shield come from? Well, to find the answer, we have to go way, way back in time. See, here's the crazy part. This mutation has absolutely nothing to do with HIV. It didn't evolve to fight this virus. It's more like a genetic echo, a shield that was forged in the fire of some other ancient plague that just so happens to be the perfect weapon against a modern one. And the first clue to what that ancient plague might have been is looking at a map. This mutation is basically only found in people of European descent. You hardly ever see it in people with African, Asian, or Native American ancestry. And as you can see, it's most common way up in Northern Europe. That kind of pattern isn't an accident. It's a clue. It's a footprint left behind by history. All right, so here's the theory. Scientists think this mutation popped up maybe 2,500 years ago. And for it to get so common in that part of the world, it had to have been a massive advantage against something that was killing a lot of people. One really cool idea is that the Vikings helped spread it all over Europe during their raids. But what was it protecting them from? Well, the top theories point to other massive killers like the Black Death or maybe smallpox. The idea is simple. If you had this mutation, you survived the plague. If you didn't, you didn't. Survivors passed the gene on, and over centuries, it became more and more common. And all of that ancient history leads to one stunning fact today. 1%. About 1% of people with Northern European roots have 2 copies of this mutation. Think about that. That means 1 out of every 100 people from that part of the world are just born immune. They literally cannot be infected by most strains of HIV. It's incredible. So now we have the blueprint. We know the exact genetic code for immunity. The problem is, Timothy Ray Brown's cure, the whole wiping out the immune system thing, is way too dangerous and complicated to give to millions of people. So that led to the next big question. What if we could give someone this immunity without needing a donor at all? And this is where things enter the realm of true modern medicine. Forget finding a 1 in a million donor. What if we could just take a patient's own cells, pull them out of their body, and edit them? What if we could perform a kind of microscopic surgery on their DNA to just snip out that little piece and create the mutation ourselves? Well, it turns out we have the tool to do just that. It's called CRISPR-Cas9. You've almost certainly heard of it. It's a huge deal. People call it molecular scissors, and that's basically what it is. It's a technology that can scan through all of your DNA, find one specific gene, like the CCR5 gene, and make a perfect, precise cut. It's the key that lets us literally rewrite the code of life. So how would this actually work in practice? Well, the game plan is pretty straightforward. Step 1, you take some of a patient's own stem cells, the cells that create their entire immune system. Step 2, you take those cells into the lab and you use CRISPR to snip their CCR5 gene, basically giving them that delta 32 mutation. After that, you just grow a bunch of these new edited cells. And finally, step 4, you put those genetically modified HIV-proof cells back into the patient. They then go to work building a brand new immune system from the inside out, a fortress that HIV can't penetrate. So think about the journey we've just been on. We started with one man's unbelievable cure, which led us to an ancient genetic shield passed down from plague survivors, and that in turn led us to a futuristic technology that can copy it on demand. This is bigger than just one disease. What we're looking at here are 2 totally different paths to the exact same result. On one side, you've got natural immunity. It's a lucky break, a historical accident, a winning ticket in a genetic lottery from thousands of years ago. But on the other side, you have engineered immunity. It's not an accident. It's a deliberate, precise, scientific achievement. One is a gift from our ancestors. The other is a tool we built for our future. And make no mistake, this isn't just a cool idea on a whiteboard. The potential here is absolutely massive. Clinical trials are happening right now. The research is showing that CRISPR could actually be a cost-effective, reliable way to do this. We're talking about taking a super rare genetic gift and finding a way to make it available to everyone who needs it. And all of this leaves us with one final mind-blowing question. We started this whole story talking about one virus, HIV, but the tool we figured out how to use along the way, the ability to literally edit our own DNA, that's the real story. If we can build a shield against this one virus, what's next? What other genetic diseases could we just delete? What does it mean for us as a species when we can start directing our own evolution? The story of this one mutation, it turns out, isn't just about HIV. It might just be the start of a whole new era for humanity.